Much, Nina, and thanks a lot, everyone, for attending this. This is supposed to be, a very brief overview of what we have been doing as part of our COVID-19 imaging research over the past year and a half. I'll go over some of these, but if you're interested in talking about some of these in detail, we could definitely touch base offline and talk about these. Anyway, so my talk today is titled The Virus in the Details, Discovering Subtle COVID-19 Visual Features on Chest Radiographs. So before I go into some of the details of what we have been doing, I'd like to talk a little bit about the research directions of our lab. So the research theme of our lab lies at the intersection of machine learning and image analysis, where our focus is on biomarker discovery via machine learning and multi-scale data fusion. So in order to quantitatively characterize the underlying disease heterogeneity, we have been working on the development and validation of techniques such as radiomics, radiogenomics, and radiopathomics. So these approaches have been developed across different domains, including brain, breast, and lung cancer in the context of disease diagnosis, prognosis, and assessment of response to different kinds of treatments. So the different kinds of modalities that we work on radiological modalities, digital pathology, and so on. So here's a shout out to our department. So we actually worked day in and day out for over a year to put together this imaging data commons, these Stony Brook imaging data commons for COVID-19. So in fact, some of this a limited data set that has been collected as part of this effort is now public on TCIA. So we have not just imaging data, but also clinical data available for more than 1,300 COVID positive patients available on TCIA. This was made public last month. So like I said, currently this database houses data of more than 9,000 patients with different data elements, including images, clinical notes, lab data, outcomes, so on and so forth. This was a collaborative effort by multiple people from different branches. So we have faculty, interns, fellows and residents, and also medical students across Stony Brook who worked on this effort. So we received the seed grant right before summer in 2020, and this was a COVID-specific grant. The co-PIs on this grant were Dr. Dimitrios Samaras and from computer science, and Dr. Elaine Gold from radiology. So as part of this work, we have been involved in three core pieces of research. So first is on developing radiomics and deep learning pipelines to predict clinical outcomes in COVID-19. Second, developing frameworks for prediction of progression in COVID-19 using temporal chest X-ray scans. And the third is disentanglement of structure and texture in lung X-rays, chest X-rays. So here are a list of a few of our publications from the COVID imaging work that we have done so far. So the first piece of work here, like I said, is outcome prediction using radiomics and deep learning on chest X-rays. So radiomic features, for those who are not aware of this, are subvisual yet quantitative features that we can compute from different radiological modalities, be it CTs, X-rays, MRIs. And these are handcrafted imaging descriptors that can characterize the local and global heterogeneity in medical images. So we developed a pipeline where we take in chest radiographs, and then we have a few pre-processing steps that I'll discuss in the next couple slides. And once we have the regions of interest, we use these handcrafted features, we extract these features, and we have also developed a pipeline to fuse these features with non-imaging clinical data. And this was a deep learning pipeline. And once we do that, we sort of explore how these different features can help us predict outcomes such as need for mechanical ventilation or even mortality. So like most medical image analysis applications, this also needed some domain-specific pre-processing that you see on this slide. So first, as you can see, chest X-rays are extremely noisy. So the first step would be obviously to figure out where the lung fields are. So we trained a deep learning algorithm to figure out where the lung fields are. And as you can clearly see, we have these lung fields extracted. We are excluding the heart region from here. And the other thing that was interesting here was a lot of, right when the pandemic started, there were a lot of images, a lot of papers that started coming out, especially from Asia. And a lot of these initial papers, they took the entire image for their analysis. So what happens if you do that is, a lot of the regions that your machine learning algorithm learns its signals from might be, regions outside the lung, or they might also be from regions where you have, let's say, artifacts such as EKG leads and other wires, pacemakers, and whatnot, right? So we want to make sure that we remove these noisy artifacts before we can extract imaging measurements from these regions. So what we did was, in addition to extracting the lung fields, we also trained an algorithm to segment out the different artifacts. And once these are segmented out, we learn our imaging features from the pure lung parenchyma. So the next pre-processing step was histogram adjustment. So we had images, so our initial batch of images was from Stony Brook. And when we trained our algorithm and tried to test it on another data set that was collected from Newark Beth Israel Medical Center, we found that the algorithm did not work as expected. Now, when we went back and saw what exactly was going on, we took a step back and we sort of figured out that the machines were obviously different, but the intensity of the images at the same windowing level were quite different for Stony Brook data and the data from our collaborating institution. So we designed a simple histogram adjustment algorithm. And once we did that, you see the results here. So these are before histogram adjustment, and this is after histogram adjustment. And we can clearly see that the images from our collaborating institution is brought to a similar sort of a distribution as the Stony Brook data. So the unmet clinical need here was to see if baseline chest X-rays can provide information regarding disease progression. And for this particular case, we were looking at outcome prediction. And this was a multi-site study. We had more than 300 cases from Stony Brook University and close to 65 cases from Newark Beth Israel Medical Center. So we trained machine learning models using baseline chest X-ray scans, which were acquired immediately after presentation of and confirmation of COVID-19. And we had a fusion of deep learning and radiomic models, and our final sensitivity on a test set was 0.90 with a specificity of close to 0.7. So what you see on the right here are salient regions that the algorithm learns its information from. And what we can clearly see here is that most of the regions that the algorithm is focusing its attention on is actually the regions where you have the COVID infiltrates. So you see the left lung here, so it's flipped. So you see the left lung here. So the lower part of the left lung is where you see a lot of infiltrates. So do you see such infiltrates in the middle part of the right lung? And we see that our saliency maps actually pick up some of these regions when you look at it in a post hoc manner. So the second piece of work that we focused on was to look at temporal scans, temporal imaging scans for COVID-19 patients. So none of the previous works before this had jointly exploited the spatial distribution within the images and the temporal distribution across the different time points. Now, as you can see here, this is the same patient, and we can see the COVID infiltrates progressing over time. So this was the first images upon presentation. We can see some of the infiltrates in the left lung and the right lung, and we can clearly see the consolidation sort of become denser in B and C. You can see this really hazy pattern. And when you look at image D, you can see that some of it has resolved a bit. So clearly it's not a monotonic increase or a monotonic decrease. At some point it peaks and then goes down, or it goes down and then goes up. So we wanted to build a system that could sort of track the evolution of the COVID infiltrates over time. So this would essentially help us, provide imaging evidence of, disease improvement or disease degradation on sequential imaging data. So for this, we had close to 100 patients, and this comprised more than 600 scans. And we essentially had the radiologists go in. We first had a deep learning algorithm that segmented out the lungs, like in a previous project. And then our collaborating radiologists essentially went in and provided severity scores ranging from 0 to 2. So 0 means that there were no infiltrates at all. 1 means medium level of infiltrates, and 2 means heavy infiltrates. So for all these 600 odd scans, they went in and they provided, so these were 3 radiologists who did that in a consensus fashion. And then we obtained all the severity scores for all these different chest X-ray scans. So the goal here was to use or to model the evolution pattern of the disease over the first, let's say, n days, and predict what will happen on day, let's say, n+5 or n+10. So for this, we designed a CNN LSTM based network. So we had both a spatial, module and the temporal module, which would essentially not just learn spatial information of the disease infiltrates, but also remember what happens on day 1, using a memory unit, take it forward to day 2, and so on and so forth. So using this approach, we also had a baseline where we used a simple transfer learning based approach, and we could see that our approach could outperform most of the baselines and different variants in ablation studies for most of the different lung zones. So essentially we had 6 lung zones, the lower, middle, and the upper zone for both the lungs. So in a follow-up approach, we wanted to design a module that would explore the similarity between adjacent regions. Now, what I mean by that is, when you look at the COVID infiltrates, we sort of observed, and our radiologists, collaborating radiologists, observed that there was a similarity of these infiltrate patterns between these neighboring zones, and sort of a lower similarity or a more dissimilarity of these patterns in faraway zones. So let's say, take an example of the right lung here. So we can see that the middle zone, the infiltrates are more similar to the infiltrates in the upper zone or in the lower zone, but the infiltrates in the uppermost zone and lowermost zone are sort of less similar to each other. So we designed a module that would capture the adjacent, spatial similarity between the different zones of the lung. So in order to do that, we designed a multi-scale gated recurrent unit encoder with a correlation module. So this correlation module was the important piece of this puzzle here, because this correlation module essentially looks at how similar or how dissimilar the information is, between the different lung zones. So using this, in fact, helped us achieve a better performance in predicting the severity at a later time point. So what I'm showing here is an example of the outputs that we got from the left lung, the upper zone, and the right lung, upper zone. And we can clearly see that our accuracy, our precision, and recall are mostly higher for our approach as compared to the baselines and also the different variants. And the different variants that we considered here was, without the correlation module, or without the we had the spatial and the temporal, without the spatial network that I showed in the previous slides. So the other thing that we noticed here as part of an observation was that in the disease course of an individual COVID patient, the in-lung texture pattern changes while the lung structure remains almost the same. So thus we assume that the in-lung texture conveys important messages about the lung disease and the outcomes. Now, as you can clearly see here, we have the disease patterns. So the first one is the disease on day 2, and the second one is the disease on day 23. So we can clearly see that the infiltrates are at a different concentration or a different level of infiltrates on day 2 and day 23, but the lung structure is similar across both these days. Now, the observation here is that the in-lung texture correlates with infiltrate levels, while the lung structure remains unchanged throughout the disease course of an individual patient. So what this tells us is the in-lung texture is essentially reflective of disease severity. So what we decided to do was, now since we sort of, based on the different observations from our radiologists, we found that the texture is essentially that dictates what the level of infiltrate is. Can we design a module, or can we design a framework that can help us disentangle lung structure from lung texture? Now, if we can do that in an unsupervised way, we can then learn what these textures are, and then are these textures different across different pulmonary disease manifestations. So in order to do that, we developed a deep learning framework. It's a generative approach to disentangle structure from texture in lung chest X-rays. And this was called the lung swapping autoencoder. And this was for representation learning. So essentially what this does is it learns, it has a texture encoder and a structure encoder, and it encodes them separately. And when it decodes the representation, we can actually use the structure from one lung and the texture from another lung, or texture from the first lung, structure from the other lung, and do a mashup of the structures and textures to generate new images and to learn texture patterns and structure patterns from different chest X-ray scans. So without going into the details of how it actually does this, I'll show you some examples of the swapping results. So here we have the first image on the left. So let's say we take the structure of the first image, if you look at the upper branch, and the texture of the second image, and we generate this hybrid image. You can clearly see the first hybrid image has retained the structure of image 1 and the texture of image 2. So if you can see here, in the first image here, we see that the COVID infiltrates, which are present here, have been removed because it takes the texture from the second image. Similarly, if you look at the lower branch, we have taken the texture of the first image and the structure of the second image. So essentially, you can see this image, this hybrid generated image here. You can clearly notice that the structure of the first image is retained, which essentially you don't have any COVID patterns here, but it takes the texture from the second image and hallucinates the COVID patterns on this image right here. So this can help us achieve two things. First, you can learn these texture representations or these structure representations and then use them for downstream applications. And the second is you can use this as a generative model to generate new images. Let's say you can take images with healthy lungs and take the structure of the healthy lungs and hallucinate what a disease might look like on images like this. So this was recently accepted for publication in MICAI, and I believe will be presented tomorrow. So after the lung swapping autoencoder was trained, we applied to perform two downstream tasks. First, we used the texture encoder in the LSAE, or the lung swapping autoencoder, to do pulmonary disease classification on a very large chest X-ray 14 dataset. Now, this dataset has close to 100,000 images of, with different kinds of diseases such as effusion, infiltration, pneumonia, pneumothorax, and so on. So we use this method to encode the structure representation and then use it for downstream classification of these different diseases in the chest X-ray 14 dataset. Now, you can clearly see, so these are the different methods that we compared our method against. So other methods had a lot more parameters as compared to ours. and our method outperformed most of the other methods, and it came close to the inception V3, but our method had less than 50% of the parameters that inception V3 uses. So the second task is on outcome prediction on a data set, which we call the COVOC data set. So this was a data set that we collected here at Stony Brook University, and also our collaborating institutes at university hospitals in Cleveland and at Rutgers. So using our method, we could show that LSAE achieves superior performance, which demonstrates that better disentanglement leads to better performance in outcome prediction tasks. So this is the balanced error rate. So our balanced error rate is lower than inception V3 or the naive swapping autoencoder, and our mean AUC over multiple runs in a threefold cross-validation setting is higher as compared to the other methods right here. So finally, we propose a data augmentation strategy. Like I said, this can help us augment existing data sets by learning texture from a group of images and structure from another image, and then do a mashup of them. So for each chest X-ray labeled, with Y in the COVOC data set, we can generate K hybrid images by mixing the texture of this image with the structure of the healthy images that you see here. Now, by attaching the same label Y to the hybrid images, we can actually enlarge or enhance our data set by K times. Now, when augmenting the data set by just 2 times, we see how much increase in performance we get on the outcome prediction tasks. So this can actually be used to enhance the data set by as many number of times. This is a new augmentation strategy that we can use for deep learning applications when there are limited data sets. So let's say in the beginning of the pandemic, we didn't really have a lot of X-ray scans, so we could actually use something like this to augment our data set to learn signatures in a better fashion. Now, the last slide that you see here, so this essentially shows the attention of our deep learning models on the different images. What are the different salient regions that it learns its representations from, and we can clearly see that for the correctly classified cases, the arrows here represent where the COVID infiltrates are, and the heat maps represent where these signals are taken from that that the machine thinks is reflective of eventual outcome. So for the most part, we see that it sort of tallies well with where the infiltrates are. There are obvious misclassifications, as we can see in the lowermost row here. So I'd like to thank all the students in my lab, my collaborators, not just at Stony Brook, but also at other places at Rutgers and at Cleveland, and also OVPR Seed Grant that we received that made this research possible. So before I conclude, so many of the techniques that we are developing here, even though they're domain inspired, they are very much disease agnostic. So for example, some of the pipelines that we have used here have initially been developed for cancer applications and were seamlessly transitioned to COVID-19. Similarly, we are exploring how some of the methods that we have developed for COVID-19 analysis, especially the generative approaches and the other machine learning approaches for data fusion, can also be used for other precision medicine applications in other domains, not just restricted to COVID-19. So with that, I'd like to open the floor for questions. Happy to take any questions or any suggestions that you might have. Thank you.